Variant surface glycoprotein density defines an immune evasion threshold for African trypanosomes undergoing antigenic variation, presented by Phillip, Butz, Edgar, Gonzalez, and Becca Laplante. This study was done in the lab of F. Nina Papavasiliou at the Rockefeller University in New York by Jason Pinger and Shanin Chowdhury. Dr. Papavasiliou has since started a new lab at the German Cancer Research Center in Heidelberg, Germany. The study was published in Nature Communications, which has an impact factor of 11.880. The study was funded by Rockefeller University Graduate School funds and NIH NIAID grant AI 085973. Trypanosoma brucei is a type of protozoan parasite that can evade the adaptive immune response. It does this by undergoing variant surface glycoprotein, or VSG, coat replacement. Total coat replacement takes several days. Its genome contains a large repertoire of VSG genes. T. brucei is vulnerable to early IgM antibodies for a limited time. Early IgM antibodies can lose the ability to clear T. brucei at early stages of VSG coat replacement.
Trypanosoma brucei is an extracellular protozoan parasite that is able to evade the host immune system by switching its variant surface glycoprotein, or VSG, coat that completely covers its surface. Trypanosomes have about 2,000 VSG genes, but are only able to express one VSG gene at a time. The VSGs are densely packed on the surface of trypanosomes, and they number in tens of millions. The host is able to initially mount a robust VSG-specific antibody response against T. brucei. However, it is the few that switch their VSG coat that are able to evade this host immune response. This results in a cycle of parasitemia that occurs in 5 to 8 day intervals. Essentially, the host has to remount its antibody response as if it's seeing the trypanosomes for the first time. During VSG coat replacement, the parasites can show both pre- and post-switch VSGs on their surface. The pre-switch VSGs are still vulnerable to the initial antibody response. The post-switch VSGs are no longer vulnerable to the initial antibody response and must wait for the second antibody response. Once a trypanosome has started replacing its VSG coat, it must completely replace its VSG coat and will not stop halfway in between. Complete VSG coat replacement takes several days. The author's main hypothesis of this paper is, what are the dynamics of VSG coat replacement and how does this impact IgM-mediated immune evasion? In order to measure the rate of VSG coat replacement, the authors used the transgenic T. brucei line delta 70. The delta 70 line was then engineered to be sortaggable by replacing the wild-type VSG2 gene for one that can have an N-terminus, in this case called VSG2-STA. This has been engineered to have two alanines on the end of it, thus giving it its N-terminus. They use the delta 70 line because it has a high rate of VSG switching in the presence of doxysilane. Doxysilane induction causes a double-stranded break to occur near the active VSG gene. This causes a high percentage of the population to switch their VSG coat exactly once. The doxysilane-induced delta 70 line has a switching rate of about 5.2% of the population compared to about 0.4% of the population when non-induced. Now, what is sortagging? Sortagging is the process of attaching a fluorescent molecule, fluorophore in this case, to a sortase recognition motif and then incubating the protein that you want sortagged, in this case our VSG2-STA, with the sortase recognition motif ligated to a fluorophore and sortase. The sortase will covalently link the fluorescent sortase recognition motif to the N-terminus of the protein, hence why the authors had to engineer the VSG2 to have an N-terminus. Supplementary figure 1C shows that the sortase, the fluorescent tag, and both in combination have no significant impact on the population growth. This was measured by mean fluorescent intensity of the intracellular fluorescent stain, CFSE, which dilutes as a direct result of cellular proliferation, and is used as a cellular proliferation metric for this exact reason. Supplementary figure 1D shows the difference in VSG2 loss in DOX-induced and non-DOX-induced VSG2-STA proteins. No significant difference was reported between the wild-type VSG2 loss and the VSG2-STA protein loss. Figure 1A shows the rate of population VSG switching as measured by flow cytometry. Although they did use the delta 70 line that has a higher rate of switching, only about 5.2% of the population switched their VSG code. In order to quantify VSG loss, mean fluorescent intensity from flow cytometry data was converted into molecules of equivalent soluble fluoroform, or MESF, as this measure represents the actual number of VSGs tagged per cell. In figure 1C, the left-hand side shows the mean number of VSGs per cell, and the right-hand side shows the total SOR-tagged VSG in the population. This was calculated by subtracting the MESF from the background MESF and multiplying by the total number of cells. Figure 1D shows the quantification of VSG loss in switched and non-switched cells, both doxycycline-induced and non. They found that there was no difference in VSG loss between these populations, indicating that VSG half-life is equivalent across switchers and non-switchers. Supplementary Table 1 shows the equations that the authors were able to create from the nonlinear regression analysis from the flow cytometry data in Figure 1D. From there, they were able to calculate the effective half-lives for the VSGs in either switchers or non-switchers in both DOX-induced and non-induced populations, as shown in Figure 1E.
3 dual expressor clones were produced, each with unique UTRs. To determine the level of VSG2 expression in the dual expressor clones, the 3 clones and wild-type VSG2 expressing trypanosomes, which are the positive control, were stained with anti-VSG2 and analyzed using flow cytometry. The fluorescence signals of the dual expressors were shown as a percentage of the fluorescence seen in the wild-type trypanosomes, which are assumed to have a full VSG2 coat. The 3 dual expressor clones each contained unique UTRs and were named DE1.3, DE7.6, and DE11, each clone expressing 1.3%, 7.6%, and 11% VSG2 compared to the wild-type trypanosomes, as shown in the graph and in the table in panel B. It is possible for the fluorescence intensity to become attenuated at higher VSG2 levels if not enough anti-VSG2 is used. Thus, the authors tested increasing concentrations of anti-VSG2 to stain wild-type VSG2 trypanosomes for flow cytometry and verified that the concentrations of anti-VSG2 used in the experiments, which were shown by the arrow, represent a saturating concentration of the antibody. Furthermore, the authors verified that the expression of VSG3 is unaffected in the dual expressor clones by staining the trypanosomes with anti-VSG3 and analyzing them using flow cytometry, shown on the right. The fluorescence intensities of the dual expressors match that of the wild-type VSG3 trypanosome positive control, demonstrating that VSG3 expression remains unaffected in the dual expressor clone. Using the same setup, the authors also tested the expression of VSG2 over time in wild-type VSG2 trypanosomes, wild-type VSG3 trypanosomes, and the 3 dual expressor clones. In this experiment, the wild-type VSG2 expressing trypanosomes represent the positive control, while the VSG3 expressing trypanosomes represent the negative control. After staining the trypanosomes with anti-VSG2, flow cytometry was used once again, and the fluorescence intensities of each sample were compared daily over a period of 14 days. The results showed that the expression of VSG2 remains constant over 14 days in all 3 dual expressor clones and in the wild-type trypanosomes. These results established that the three dual expressor clones could be used as an experimental model to determine the influence of partial VSG switching on trypanosome immune evasion, since the three clones express different ratios of VSG2 to VSG3 constantly over time.
Figure 3 examined if there is a specific threshold of new VSG proteins needed for immune evasion to occur. A group of 49 mice was immunized against VSG2 trypanosomes, while a group of 49 mice was left untreated as a negative control. In order to achieve an IgM-only response, the mice were immunized with UV-irradiated VSG2 trypanosomes that lacked endogenous lipase GPI-PLC. This allowed for the VSG coats to remain intact after UV irradiation and ensured that the VSG coats remained composed of only VSG2 proteins. Trypanosomes are generally cleared by T-cell independent VSG-specific IgM responses. There is a small set of B cells, B1 B cells, that can be activated to produce antibodies against a specific pathogen without T-cell help. These B cells don't undergo class switching or affinity maturation, so the antibodies produced are low-affinity IgM antibodies. There are two broad classes of antigens that can stimulate a T-cell independent response: Ti1 and Ti2 antigens. Ti1 antigens are primarily composed of bacterial cell wall components, such as LPS, and bind to TLR4 on the B cell surface. Ti2 antigens are polymeric protein antigens or capsular polysaccharides. Instead of binding to TLR4, these antigens bind to the BCR as well as complement fragments C3d and C3dG. This causes cross-linking of CD21, the receptor for C3d, also known as CR2, and the BCR to stimulate the B cell to produce IgM antibodies. This Ti2 response is what occurs in trypanosome infections. Panel A shows the antibody levels at 8 days post-immunization, which were measured via ELISA. IgM levels increased only in the immunized mice, shown in red, and IgG levels were unaffected in both groups. This panel confirms that the immunization was successful in inducing IgM antibodies against VSG2 proteins, and that IgM was the antibody responsible for the results in panels B and C. Each group of 49 mice was split into 5 groups, for a total of 5 groups of immunized mice and 5 groups of non-immunized mice. 8 days after immunization, each group was then challenged with 100 live trypanosomes of one of 5 different strains: wild-type VSG2, wild-type VSG3, DE1.3, DE7.6, and DE11. Each strain had 10 mice per group except for immunized DE7.6 and non-immunized DE11, which had 9 mice per group. One mouse in the immunized DE7.6 group died due to an unrelated cause, and one mouse in the non-immunized DE11 group had high levels of IgM antibodies against VSG2 and was not included in the data. Panels B and C show the data for each group of mice for percent of mice with confirmed infection and percent survival respectively, starting at 4 days post-infection. The gray shaded area on each graph begins at the estimated time point that the initial parasite peak is cleared and antibodies against the challenged trypanosomes are being produced. This means the data in the gray area is less clear as to how much of a role the immunization is playing in the long-term infection and survival data. The data in the white unshaded region, however, can be interpreted as a direct result of the immunization against VSG2 proteins. Generally, we can see that the more VSG2 proteins present on a specific trypanosome, the better the prognosis is in VSG2 immunized mice. The immunized mice infected with wild-type VSG2, shown all the way on the right, showed a 0% infection rate and a 100% survival rate as expected. Conversely, the immunized mice infected with wild-type VSG3, shown all the way on the left, showed between 80 to 100% infection rate and ultimately a 20% survival rate, which is similar to the non-immunized results shown as a dashed line. The results were similar for the immunized mice infected with DE1.3 trypanosomes as well. These trypanosomes had only 1.3% VSG2 proteins. They showed a 90 to 100% infection rate and ultimately a 20% survival rate. The immunized mice infected with DE7.6 trypanosomes, however, showed a sharp change with about a 20% infection rate and around a 90% survival rate. Interestingly, The immunized mice infected with DE11 trypanosomes had a higher infection rate, around 75%, but it was delayed by 11 days compared to the wild-type VSG3 group. The DE11 group also had a lower survival rate, ultimately around 60%. It's interesting that these DE11 trypanosomes, which have a higher percentage of VSG2 proteins than the DE7.6 clones, had a slightly worse prognosis. This could be due to the pentameric nature of IgM antibodies, in that even though they are more VSG2 proteins to recognize on the DE11 clones, they may not be spaced correctly for the IgM pentamer to successfully bind. This could also be due to the original trypanosome population undergoing VSG switching. Despite this, the data still support the overall hypothesis of the paper that there is a small window of time in which trypanosomes that are undergoing VSG coat replacement are vulnerable to pre-switch IgM antibodies. It's clear that having a successful initial immunization plays a role in promoting survival and clearance of the trypanosomes.
In order to study the mechanisms of IgM binding to VSG that result in immune evasion during the late stages of VSG switching, the authors produced anti-VSG2 polyclonal IgM in 2 mice by injecting the mice with wild-type VSG2 expressing trypanosomes. Veronil was administered to the mice 4 days and 5 days after infection to kill the trypanosomes before they were able to switch VSGs and trigger a new IgM response to the newly switched VSGs. Polyclonal IgM was then obtained from the mouse serum. In this experiment, trypanosomes were incubated with polyclonal anti-VSG2 IgM from the mouse serum instead of monoclonal antibodies as in Figure 2. The anti-VSG2 IgM stained the DE7.6 and DE11 dual expressor trypanosomes and the wild-type VSG2 trypanosomes with increasing fluorescence intensities observed in trypanosomes expressing higher concentrations of VSG2. The D1.3 trypanosomes demonstrated little to no fluorescence intensity. Wild-type VSG2 expressing trypanosomes were used as a positive control in this experiment, while wild-type VSG3 expressing trypanosomes were used as a negative control. To verify that VSG2 was indeed expressed in the D1.3 trypanosomes, the 3 dual expressor and 2 wild-type trypanosomes were stained with fluorescent monoclonal anti-VSG2 IgG and analyzed using flow cytometry. The fluorescence intensity increased for all trypanosomes expressing VSG2 in comparison with the wild-type VSG3 expressing trypanosome used as a negative control. These results demonstrate an increase in fluorescence intensity proportional to VSG2 concentration in the trypanosomes. The authors devised a model that can explain the mechanism of IgM binding to VSG that resulted in immune evasion threshold at low concentrations of pre-switch VSG. IgM makes pentamers that form low-affinity interactions with the antigen and thus depends on multiple antigens binding to one IgM molecule for long-term binding. The FAB arms in IgM cover an estimated 40 nanometer area. The VSG code on trypanosomes is modeled as a dense code of VSG homodimers, with a spacing of 5.8 nanometers between homodimers. Thus, the authors inferred that an IgM molecule can bind to 61 different VSG homodimers that are concentrated together in a 40 nanometer area on the trypanosome cell surface. According to the model, if a trypanosome has a pre-switch VSG percentage higher than 7.6%, IgM can establish contact with multiple VSG homodimers on the trypanosome surface, as indicated on the 3 hexagons on the left in panel C, which show that VSG code would pre-switch VSG at different percentages. As shown in this model, trypanosomes expressing only 1.3% pre-switch VSG will make contact with less than 2 different VSG homodimers. Since a single IgM molecule cannot bind to multiple VSG molecules at concentrations less than 1.3% pre-switch VSG, prolonged binding of IgM to VSG code does not occur, and any interactions are fleeting, which explains the fact that these trypanosomes were able to evade the IgM-mediated immune response in the immunized mice.
The overall question investigated by the authors of this paper was, what are the dynamics of VSG coat replacement, and how does this impact IgM-mediated immune evasion of T. brucei? They hypothesized that T. brucei is vulnerable to pre-switch IgM antibodies for an amount of time, while T. brucei is undergoing VSG coat replacement. Figure 1 established the rate of VSG protein switching. Figure 1 found that the rate to be about 4.5 days for a full coat replacement, with about 99% of the coat proteins replaced in the first 29 hours. Figure 2 established successful creation of dual-expressing, or DE, T. brucei strains with different levels of VSG coat proteins. The authors used two main proteins, VSG2 and VSG3. VSG2 proteins represented pre-switch proteins, and VSG3 represented post-switch proteins. Figure 3 immunized mice against T. brucei VSG2 proteins and infected mice with T. brucei DE strains created in Figure 2. Trypanosomes were vulnerable to pre-switch IgM antibodies against VSG2 until roughly 99% of the VSG2 coat was replaced. Figure 4 examined the effect of VSG protein density on the ability for an IgM pentamer to successfully bind to T. brucei. This figure determined the average number of pre-switch VSGs that would be within reach of an IgM pentamer, and the protein threshold for IgM to make successful antigen contacts. Greater than or equal to 7.6% pre-switch VSGs allows for IgM pentamers to make multiple antigen contacts on a VSG coat. The authors successfully answered their overall questions. They determined that T. brucei is still vulnerable to pre-switch antibodies up until roughly 99% of the pre-switch proteins have been replaced. They concluded that this is dependent on the physics of the IgM pentamer and how many antigen contacts it can make on a VSG coat.
Previous studies have shown that antibodies are the only immune effectors that can recognize a specific VSG and help clear trypanosome infections. Due to the high VSG switching frequency observed during trypanosome infections, it is assumed that the antibody class that is produced initially will be the one that clears the infection, which is why the authors focused on IgM for this paper. However, IgM deficient mice infected with T. brucei display mild parasitemia progression compared to B cell deficient mice that produce no antibodies and displayed severe parasitemia after infection. While the authors of that particular study concluded that IgG is more responsible than IgM for trypanosome clearance, the authors of this paper argue that it was mediated by IgD instead, that was generated to compensate for IgM deficiency. Therefore, our hypothesis is that IgD and IgG, along with IgM, participate in clearance of trypanosomes during natural infections in mice. In order to test this hypothesis, we would infect wild-type mice and mice deficient in IgM, IgD, or IgG with T. brucei and observe the mice for 22 days for survival, just as the authors did for Figure 3 in this paper. We would also compare the parasitemia between wild-type mice and mutant mice by identifying the trypanosomes in mouse blood smear samples using a microscope. If the IgM, IgD, or IgG deficient mice displayed lower survival and higher parasitemia compared to the wild-type negative control mice, this would indicate that the antibody class in question is necessary for an effective immune response against T. brucei. If IgD or IgG are found to be relevant during natural infections with T. brucei, the same studies described in this paper could be applied to determine the respective immune evasion thresholds, just as the authors did with IgM.
Thank you, everyone. This concludes our presentation. Feel free to ask us any questions on the Blackboard discussion board. We'll be answering questions until Friday, May 8th.